All right, time to go teach. Hello and welcome to this next episode of Tutors, continuing the cell and continuing cellular respiration. And today we are going to be looking at the link reaction and the Krebs cycle, which is also called the tricarboxylic acid cycle, or the TCA cycle, or the citric acid cycle. Targets for the day are going to be, first, we're going to describe the link reaction and the Krebs cycle. And then we're going to identify the important products and the important reactants of these two different stages. Well, without any further ado, let us look at what's going on with these things. First off, the link reaction and the Krebs cycle, they take place in the mitochondrial matrix. So when we think about the mitochondria, remember it had the two membranes. It has an outer membrane and then it has an inner membrane, which is highly folded. And there's space within that inner membrane, and that is where the matrix is. It requires the presence of oxygen. We will not have the link reaction taking place or the Krebs cycle taking place without oxygen being present. The input is pyruvate, 4 NAD pluses, 1 FAD, 1 ADP, and 1 phosphate group, recognizing that those values are going to be multiplied by 2 because each glucose, when it undergoes glycolysis, produces 2 pyruvate. And so we're going to end up with 2 link reactions and 2 Krebs cycles occurring per molecule of glucose. And that also means that our output would also be multiplied by 2. So per pyruvate, we'd only make 3 CO2s, 4 NADHs, 1 FADH2, 4 hydrogen ions, and 1 ATP. But per glucose, we would multiply that by 2 to be our full 6 CO2s that are produced, and we are going to end up producing 8 NADHs, 2 FADH2, 8 H plus ions, and 2 ATPs per glucose. Let's look at the location of where this occurs first. It is going to be inside that inner matrix. The pyruvate is going to be decarboxylated, which means we are going to be removing CO2 from this molecule of pyruvate. As the pyruvate is decarboxylated, it's also going to provide energy to form NADH and a hydrogen ion. We're going to take in an NAD plus, and the CoA is going to bind along with the NAD plus coming across, and their energy of the carbon dioxide being released is going to provide us with the energy to build NADH and a hydrogen ion, as well as what is called acetyl-CoA. The carbon dioxide can just diffuse away, going from a high concentration to a low concentration passively through the membrane. The acetyl-CoA, that is going to be crucial for the beginning of the Krebs cycle. That is going to be part of the start that we are going to require to input into the Krebs cycle. Here's how it's going to work. First, the Krebs cycle, which, as I said before, is called the tricarboxylic acid cycle, which is where TCA comes from, or the citric acid cycle. The first step is that the acetyl-CoA and a molecule that is a 4-carbon molecule called oxaloacetate is going to end up bonding together, forming a 6-carbon molecule. The coenzyme A is going to be removed, and now we end up with a 4-carbon molecule, which, as we break this molecule down, we are going to release both CO2 and we are going to form NADH along with hydrogen ions. Releasing that CO2, we have formed the NADH and a hydrogen ion. That CO2 can then diffuse away. This is going to happen again as the 5-carbon molecule is going to get broken down even more. It's going to be decarboxylated, and then we are going to end up with, again, some more NADH and the hydrogen ion. The CO2 is going to be able to just diffuse right away. We're not paying attention to the actual structure of this 4-carbon molecule. But as this 4-carbon molecule changes shape and interacts with enzymes, it still has more energy that can be released. And that changing of conformational shape is going to be what gives us the power to not only build an ATP, but it's also going to be what provides the energy to form another molecule. From FAD, we are going to make FADH2, which is another energy-carrying molecule, much like NADH. And we're going to be able to form another NADH and a hydrogen ion, all from this 4-carbon molecule changing shape and interacting with enzymes. Our final 4-carbon molecule is oxaloacetate, which means that it can then bind with another acetyl-CoA, and we can start the Krebs cycle all over again. But at the end, we have produced 4 NADHs, 4 hydrogen ions, 1 FADH2, and 1 ATP. And that was using only the input of 1 acetyl-CoA. And we must remember that every glucose is going to provide 2 pyruvates, which means we would have had 2 acetyl-CoAs. And that means we can multiply this by 2 to see what the full products of the Krebs cycle would be for 1 glucose. The NADHs and the FADH2s are energy-carrying molecules that will be used in the final stage of aerobic cellular respiration, which is called the electron transport chain, which is these enzymes right here, which occur on the inner membrane of the mitochondria. In summary of the Krebs cycle and the link reaction, what we got is that they are going to occur in that inner mitochondrial matrix, and they are going to produce only 2 profit ATP per glucose. But that's not the only things that the link reaction and the Krebs cycle produce. They also produce the inputs that will be used for the electron transport chain, and that would be the NADH, the FADH2, and the H+ ions. Those are crucial for the final stage of aerobic cellular respiration, which we will talk about in our next video. Until then, be awesome, stay awesome.